Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Anna Lindstrand, who's professor and consultant in Clinical Genetics and Genomics at the Karolinska Institute and Karolinska University Hospital, where she also directs the Clinical Genetics and Genomics Diagnostic Laboratory and leads the Rare Disease Research Group. Anna has been at the forefront of pretty much all of the waves, I think, in advances in genome sequencing, from cytogenetics to whole genome sequencing, and we're going to talk a lot today about long-read sequencing as well. So Anna, thank you so much for joining me and taking the time today.
Thank you for asking me to join. I'm super excited about doing this.
You have a very, I think, modern and sophisticated clinical genetic setup. So I want to start actually with just asking the algorithm, basically, of let's suppose that I'm a patient in your clinical genetics catchment area. How do you think about whether to use whole exome, whole genome, long-read sequencing, reflex into some of the other genomic technologies like RNA-seq? I recognize this is a really broad question, and I just want to get a sense of if it's a rare disease or cancer, we do this. If it's something else, we do that, and we can take it from there to try and understand how you see that evolving.
Yeah, so at the Karolinska, we were really fortunate to start very early on with whole genome sequencing, pretty much at the same time as Genomics England was started back in 2015. We started doing clinical genomes, but then we were able to kind of scale up steadily. So last year, I think we did like 6,000 clinical whole genomes. So for rare disease, that is really the first line test at this point. Most of the referrals will come from other specialties where they basically send a referral and a blood sample, and we do the genome. Sometimes it can be complemented with a short read RNA sequencing analysis, also clinically. And we are currently having very advanced implementation projects when it comes to long read. And I think at this point, we have a validated long read genome that we can use as a follow-up test, but not as a first-line screening test. So when it comes to cancer, I would say that the standard in general would be more of a panel approach. And in my lab, we do the hematology, and then solid tumors are done through the pathology department. So we have at the national level organized ourselves within the Genomic Medicine Sweden project to try and really coordinate this across a regionalized healthcare system. So we have very broad gene panels that would apply for like a myeloid leukemia or a lymphoid leukemia that would go in as a first-line test. But we also have long-standing, also, I would say, implementation/research projects when it comes to early usability of genomes and on the, for instance, childhood cancer side. This is the first line test now for all childhood cancers in Sweden. They get analyzed with a genome, and most of them also with a paired normal analysis since about a year back.
You have published some really interesting recent work on the long read sequencing that you're doing. Could you talk a little bit about that work, what you're learning, and where you're seeing it improve diagnostic yield?
Yeah, so I think starting with like short read, this has been amazing for the genetic and clinical genetic community overall, just enabling so many patients to get diagnosed. But at the same time, we have somewhat painted ourselves into a corner when it comes to like the enormous infrastructures, investments that have been done, and also the computational and analysis pipeline setups are very, very focused on analyzing short read data, where I know a lot of people they compare it to like taking your encyclopedia of 46 books and then shredding them and then piecing them back together, right? So to me, I think long read is obviously better than short read. However, currently, because I mean, the reads are longer and we can get a better view of the genome, which is what we all want to do. However, currently, all our analysis setup is kind of using approaches that are more appropriate for short read. So that is a little bit of a like a 360 turnaround, right? Where you have to think about this, okay, how do we use these long reads and how do we prove that they're actually clinically useful? And now a lot of people also want us to do like health economy. And
Yeah.
I'm like, okay, but it's obvious they are longer. They give us a better resolution across the genes that we want to look at. But that's not good enough. Obviously, we need to make sure that they work in a clinical setting. They need to work on different types of DNA quality. They need to work, consistently 100% of the time. Or if they don't, we need to know that when this is the case, this scenario won't work. Also, we can't currently scale up. Like I said, last year we did like 6,000 clinical short read genomes at the Karolinska. We don't have the capacity to move them straight up to any long read platform without huge investments in new like infrastructure, which won't happen overnight. So we need to make really carefully designed implementation studies where we try these new technologies clinically and also learn how to analyze the data in a clinical setting. Because currently, sequencing is really, really easy. And I think the real frontier comes in the interpretation step.
Yeah. With long reads, what are you seeing in terms of improvement in diagnostic yield? Is it pretty uniform across different populations? And I guess it's primarily big or small CNVs that are really hard to resolve from short read sequencing. I'd love to understand what the lift that you are seeing from it is and where it's primarily coming from.
So we did one study. We did 100 kids with neurological disease. So it could be neurodevelopmental, neuromuscular, where they basically came in for a short read singleton genome. And we have a very good, again, short read singleton genome because we have a huge database and we worked a lot of our kind of analysis strategies on the computational side. And then we have a ton of super talented interpreters, both molecular biologists and medical doctors that really squeeze the clinical yield out of this data. So that said, I think most of the known genetic causes of neurodevelopmental and neuromuscular disease have sort of been discovered with short read or in the pre-short read era. That means that we don't have a lot of causes that warrant long read sequencing yet. A few years from now, we're going to have those. But right now, they're not really there. So when it comes to like yield is also a little bit of a definition. What is this? But overall, like, is there any type of a signal in the data that we're looking at? We don't get a lot more with long read. There can be the occasional small CNV, but in general, what we get is the completeness of the analysis. And here the added value is on the structural variant side, the full complexity of the structural variant, as well as the STR resolution and genotyping. And I think with the STRs, we really will also see an added yield because we can get these interrupting repeats. We can get a really, really nice, clean, exact repeat size, etc., which will be extremely helpful clinically. But overall, we get a much more complete analysis with the long reads compared to the short reads. We also can phase the data across the recessive genes. So in 100, we have 2 to 3 where we really have these compound heterozygous variants that are, immediate in the long read data. But also we have a couple of others where the variants don't phase that we can exclude that these are not recessive. So there's a lot of, I would say around 15%, we had a clear added value with long reads already with a very kind of easy analysis and not really adding in methylation at all.
Interesting. Yeah. That's another. It sounds like part of what you're saying as well is that we need to get to a bigger scale on methylation and in terms of number of cases worldwide that are being sequenced on long reads to understand the true yield. I also wanted to ask about RNA sequencing and proteomics. You've published using each of those tools as well as I suppose either helping you to interpret what you're seeing in the short read or maybe as a second line where the short read isn't giving you an answer. Can you talk a little bit about how you're using those two technologies?
So the nice thing with DNA is that it's very stable and you can take a sample and obviously you can have sizes and the varies between tissues and so, but overall, you have your DNA, you can even keep it in room temperature for quite some time, without having any issues. So RNA, in it's a mess. Its genes are expressed differently across tissues, and if we get a blood sample, we can only query around 60% of the known OMIM genes. However, that said, a lot of the variants are actually easier to interpret when you look on the RNA side. We do see a lot of low-ranked splicing variants that are popping up there and can be confirmed by adding RNA to the kind of genetics toolkit. Some, would even argue, even way back, that maybe, if we did RNA instead of DNA, we would have a higher yield overall because even for like clearly monogenic diseases, we find a little bit more. I'm not sure that's true today with all of our databases and the way we filter variants and so, but I think it is a very important tool to add in certain instances. And maybe we should do it, in a more standardized way that if we have a negative genome and the patient, warrants further analysis, we always add on an RNA analysis. And it could be from blood, it could be from fibroblasts, it could be, in some instances, even maybe we should do this rapid induction into neurons because some genes are only expressed in that tissue, and that can give us added clues as to what genes are involved.
Yeah.
Proteomics, I would say, is further from clinical diagnostics at this time. It will really need to prove itself. There will be instances, I think, where maybe this will be applied quite rapidly because that, well, in this kind of disease, proteomics will help us a lot. But on the rare disease side, it's not really there yet. I'd say that with the more and more with long read sequencing, methylation will become the next tier in the multi-omics data sets that we'll explore more clinically.
Oh, interesting. Why do you think that one will be the next tier?
So I think already, right, there's been a huge interest in these episignatures that can direct you into certain disease tracks and seem to be quite consistent between individuals. But then there's a lot of data indicating that actually quite a lot of the variants that we are finding have some sort of a methylation footprint. And we need to explore that. We need to build the databases. Maybe there will be methylation outliers. We might need to have age-specific even databases for this or not. So again, we need to understand how the methylome changes in rare diseases patients, in normal individuals, and how can we use this clinically. And likely it's going to be a similar story as the RNA-seq, that we find those methylation outliers, and when we can link them to a clear genetic DNA variant, they become the immediate functional test proving that this DNA variant is actually the causative variant in this case.
Yeah, I think that makes sense. I want to hear a little bit more about genomic medicine Sweden. I know you're co-chair of the rare disease working group. And like you mentioned, it started around the same time as Genomics England. I think there's a lot of overlap in focus areas, rare disease, cancer, pharmacogenomics. There's also novel areas where genomic medicine Sweden is focusing that other national scale programs aren't. It would be great if you could tell people who aren't familiar with it a little bit about the priorities, the goals, and what's unique about the GMS program compared to other national scale programs that they may have heard of.
Yeah, so Genomics England, I guess, was the front runner in this area, and that was really a top-down project, where Sweden is the opposite. So we're a bottom-up initiative where everyone came together, and we're at the time where genomic technology is exploding, and we need to harmonize this at the national level to both ensure quality, but also enable access, because Sweden is regionalized. But even so, we want people with rare disease, which is my main area, to be able to have access to the same frontline tests, regardless if you're born in Stockholm or in a city in the north of Sweden. So when we started, we sort of had to harmonize our, I guess, expectations and vision in a way that some centers had already. So we started with genome sequencing in Stockholm clinically in 2015, and Genomic Medicine Sweden was formed in 2017. So at the time, there was a lot of interest in short read sequencing, and some regions were investing in infrastructures, and other were more interested in maybe kind of collaborating with another center. And then over the years, we've been working a lot on the computational side, and we have a national rare disease analysis pipeline and the NF-Core rare disease pipeline, where people across the nation have been able to kind of contribute computationally on GitHub and building this together. And it really doesn't make sense in a small country, we're 10 million altogether, to build one pipeline in Stockholm and one in Gothenburg. Yeah. And it's just wasteful. And especially if we're thinking about, healthcare funded institutions. So, but on the other hand, I think with short read, people have sort of also been having to find their own way forward. But what we realized was that there was a big need for coordinating also the next step. Like, okay, we have a clinical analysis, the yield, depending on inclusion criteria, can vary from 25% up to almost, 50%. But there's so many patients still undiagnosed after that. So what we did and what we were, agreed on nationally was that we started a national rare disease multi-center study to be able to explore new technologies and share data between regions and also internationally on both unsolved and in some cases solved cases. So that has been going on since it got ethical approval in early 2019. And then we built a digital platform so patients can log in with their digital ID and consent to the study. So if you do clinical genome sequencing, you can then, after this is done, log in and donate your data to research or consent to research electronically or on paper together with your physician or genetic counselor, if that is a better fit, which I think has also helped including patients into this national study. So we have also been building I think it could be similar to the National Genomics Research Library that you guys have. It's called the National Genomics Platform, or NGP, where the idea is that then data from all individuals that have undergone clinical genome sequencing can be pooled. And for those that have consented to research, data sharing can then happen. And we can process the data there together and also use this to try new tools and develop our analysis pipelines. So this is being constructed, and I think some data is already deposited there. There's a lot of legal regulations that always need to be updated when it comes to data sharing in genomics. But this will also become our kind of portal into the federated 1 million genomes data space, etc. I think, so finally, when it comes to genomic medicine Sweden, when long read sort of entered the market and became more of a clear clinical initiative about 1.5 years ago, we all really came together and everyone was like, okay, we need to push into long read. We all need to understand more about this. What are the opportunities and possibilities in the clinical space? So we have a really fun project happening there right now.
What is, I think one of the key priorities of the program is translation. How do we improve diagnostics? How do we improve access to advanced therapeutics off the back of that? I'd love to have your perspective on it sounded like he said 2017 was when it really kicked off. So it's been 9 will be next year 10 years since that kicked off. I'm curious to hear where you've seen a lot of progress that has surprised you. And then on the flip side, what are the things that you think in the next 5 years as we get to 2030, you're really hoping we see more progress, especially on the translation front, like going from we've done the research that, to your point earlier, shows this is a useful test to we've rolled it out across the whole country and there's really meaningful impact for every Swedish citizen as a result of whatever it is that you're thinking about.
So that's a good question. I think really, I mean, again, the possibilities of genomics and working together to create impact has in itself been one of the most important tasks of the Genomic Medicine Sweden initiative. Really being able to learn from one center to the next and push diagnostics as far as it can get. That was, I guess, a little bit cliche, but it does enable us to get access to the smartest people in a specific area. I mean, if it comes to CNV interpretation from genomes, and if they build a really, really good tool for that in Lund, we can get access to that in Stockholm. If we know that someone in Stockholm is an expert on repeat expansions or disorders of sex development and these really tricky genetic results, you can get access to that in other regions as well. So I think that has been the key take home, really kind of spreading the expertise across the nation. But what's also happened over these years is that we started out as genomic medicine Sweden, and then quite rapidly, precision medicine became sort of its own thing. And to me, precision medicine is really made up of precision diagnostics plus precision therapeutics. And I'm more on the genomic medicine side, and I'm not an expert when it comes to therapeutics, but this is a giant shift in the field of genetic diagnostics where we have been traditionally very, very focused on finding these results that can result in carrier testing and prenatal decisions. And we set the bar really, really high for what variants we report out, because they really need to translate over to unaffected individuals and their decisions when it comes to family planning. But in precision medicine, we need to think about, are these results actionable for the individual we have in front of us? And this is a completely new way of thinking about genomic analysis that will impact all of us, all of the centers globally in the upcoming 5 years, I would say. We need to have new kind of standards and reporting criteria that go beyond the current ACMG criteria, where we're like, okay, this can warrant a new epilepsy drug, but we're not at the point where we know if we can offer prenatal testing just yet. So I think this is super interesting in a way for the genetics community as we're moving in that direction. The other part of the kind of enormous therapeutic potential when it comes to rare disease is that these N of a few trials are quite demanding, for the healthcare system, because we need to set up a way of enabling the patients in our country or our part of the world to come in maybe twice a month to a center, and there could be 5 individuals in all of Sweden, or in Sweden, Norway, and Denmark together. And should they then instead fly to London, or should we have these, trial hubs in different cities and so on? Here, what is starting to happen in Sweden, that we're getting these precision medicine centers, or PMCs. And here we really need it's often the pediatrician still, right? But we need the treating physicians on board, whether they're ophthalmologists or pediatricians and the experts that will actually see the patients when it comes to these trials. But we also need genetics, because we need to set the bar for which patients do we consider eligible for these trials. So it's a new, I think, world, super interesting, and maybe we'll get guided by AI in some sense here, but we're not really there yet. We have to do it ourselves first.
Yeah, it's interesting. It sounds like you're saying there are some rare, ultra rare conditions where there may only be 5 or 10 patients in the whole country, and it would be unlikely that a pharmaceutical company would set up a site in Sweden, and so we need EU-wide or global collaboration. But there are also some conditions, like I'm thinking about Usher syndrome or ATTR. There are particular variants, familial hypercholesterolemia. There's a number of conditions where they're at much higher rates, right? Are you seeing that Sweden is becoming a destination for trials increasingly for some of these conditions where there's either founder effects or other reasons why there are particular variants at a higher rate in the country?
I mean, I think we definitely should be. I'm not sure whether or not we are.
Yeah.
One of the ways I'm thinking about this, well, national genomic platform where we will eventually, hopefully, will be depositing all of our genome data, is that it sort of becomes a clinical trial ready cohort in a way. So we have really good registries in Sweden. We have longitudinal follow-up data. We have electronic healthcare data. We have advanced genomics programs. So if we have a way of finding the individuals that genetically would qualify for the upcoming, phase 2 or phase 3 clinical trials, Sweden would be a great country to conduct these trials. But we have to build a system where we can find them, because it might not even be that those genetic variants that are, well, I think for Usher and for FH, they will be reported out, but for FH, I don't even know if they are testing genetically
Yeah.
at the rate that they should be. I know that this is something we're currently discussing, and so. But for some syndromes or diseases, the variants that will be indicative of whether or not an individual could warrant participation in a clinical trial might not even be reported today, but they could be in the databases. So if we can then search them and find them that way, that could be one step forward.
Yeah, I mean, Estonia has been fairly, I think, ahead of the curve on doing recontact by genotype out of the Estonian biobank. Have there been any concerted efforts on that front, or is that something that may come in the future in Sweden?
Yeah, it has been. No, I would say we haven't been doing that, at least not at scale. I mean, we're doing some targeted reanalysis of data right now at the Karolinska, because obviously those really early genomes didn't have the computational setup that we're seeing today. I mean, but usually when it comes to like having a new potential clinical trial like we have now, right now, the MECP2 duplication syndrome, we would have to go back like specifically for that and look into, could we potentially have any suitable individuals in our region at this point, and work it from that angle. But I think if we have now a network of precision medicine centers nationally at all the university hospitals with corresponding medical universities, and maybe that's where those requests could come in.
Yeah.
And then if there are good genomic data repositories, there could be standardized way of querying that data to see if we have eligible, patients in our healthcare region.
Yeah. You have, and you talking about FH actually reminded me to make sure I touched on this. It is there's often monogenic forms, and we understand the most about monogenic forms of FH, but there's also been a lot of interesting research on polygenic drivers of high cholesterol, and almost every common complex condition. There's a group of people who don't have a clear monogenic cause, but there's probably a polygenic signal. You wrote a really interesting review that came out in January titled Moving Beyond Monogenic Disorders in Clinical Healthcare. I'd love to get your perspective on where the biggest opportunities are right now, the low-hanging fruit for, to use a cliche, that we really could be thinking about polygenic risk scores, or just considering more than one variant and using that to guide care.
Yeah, so I think polygenic risk scores still haven't really proven themselves clinically. They could be used a little bit in certain instances, but we haven't really been able to use them as this kind of determining factor whether or not, to include someone in a screening program, etc. But I think we've been so focused on the monogenic side, and the question I think we raise a little bit in this piece is anything truly monogenic or a variant in some way in a system, with the genome being a system or a specific pathway being a system? Will always the effect of that variant will always depend on all other variants in that system. So to right now, we are very, very focused on identifying those high penetrant monogenic causes in clinical genetics. And then we tend to filter out even quite interesting signals that don't fit into that particular area. And currently, healthcare is actually generating the enormous amount of genomic data. And I think it's already today more data is being generated within healthcare than in big research studies, including, I think, the population studies that are starting up across Europe due to the Genome of Europe, which are still, just the odd couple of thousand here and there. So we need to really collect these. If we call them hot VUSs or whatever, but the next level of causative variants will be in the hot VUS space. Those variants that we are already flagging and kind of clinically interpreting and assessing that this doesn't meet the threshold for clinical reporting. And then they are kind of discarded. They are not shared on GeneMatcher. We're not taking the top 5 or 10 or even 50 variants in our unsolved cases, because if we did that and shared them, we would likely see that across this particular phenotype, we have a burden of high-ranking VUSs when the clinical presentation is like this. Yeah. But we also need to think about how we define clinical phenotypes, because there we also have like a kind of a stepwise yes or no type of process with like intellectual disability or no intellectual disability. But maybe we need to look into more of a scale type of system where we look at, I'm not advocating for IQ, but IQ would be one parameter we could look at instead, or grades in school, or there are things we could look at that is not as yes or no.
Yeah, or a cognitive test, something like that. Yeah. Yeah, interesting. What are you seeing right now in terms of the value for prevention? So the reason I ask is because we're seeing the UK is investing a lot in programs like Our Future Health, that an explicit goal of that is let's build the data sets to use genetics as a preventive care. It's going to be some time, I think, before we see that delivered in the healthcare system at scale. But I'm curious to see if you're having any discussions or doing any work. We touched a little bit on newborn screening, which I think is a form of this, but in the broader population, whether you're seeing an opportunity to use this for prevention.
So we have been, over the years when we've had our genomics program, obviously we do quite a lot of trios, and when you do trios, you run the risk of sort of doing a pre-symptomatic test on a parent without them really having been able to consent to this. And it's something we've been discussing and handling. And then right now what we're seeing is that quite soon, maybe the whole idea of pre-symptomatic testing also will be turned around as we're moving into precision healthcare, right? So if there is any type of efficient drug for Huntington, the whole, pre-symptomatic test program would pretty much be ended, right? Because anyone who would like to know whether or not they have the repeat expansion and be included in these trials instead. So in a way, I feel that we want a prevention healthcare system. But maybe this is just me, I've been discussing, but I think we need to think about adult screening as an addition to the newborn screening, where individuals, for instance, when they turn 18, I mean, or 20, or what we think is an appropriate age, so it doesn't get forgotten, can go in and get tested, for instance, for BRCA and Lynch genes. At the moment, we're waiting for someone in their family to get cancer before we do the cascade testing, which feels so, I mean, backwards, right? So, but maybe to start testing newborns for breast cancer heritability doesn't make sense either. So I think there could be a possibility of having, and then I think there will be also big interest from the sort of next generation to get tested for really high penetrant recessive conditions of some types.
Yeah.
The other thing I think, and the reason this is also being discussed, is that the big part of the health economy in the genome programs we are currently running comes with pharmacogenomics. And if we really start applying that in a smart way, we could actually also save money directly. And instead of waiting for getting the side effects, actually dose our drugs efficiently from the get-go. And I think that's something that they might have built into the Estonian program. I feel like they are talking.
I think you're right about that. Yeah.
Mm-hmm.
Yeah. On the first point you made about being more proactive about screening, I definitely agree with you on that. I do see where the health economics and screening aficionados come with on that, where you can also imagine a world where you do a lot of additional screening that isn't relevant because of the penetrance of some of these variants. So there's I'm sure though a group of conditions where the data is really clear that doing earlier screening makes a ton of sense compared to waiting for, to your point, a family member to have heart failure or sudden cardiac death or cancer before kicking off the screening. I think we could be a lot smarter with analyzing not just the genetic data in isolation or that event in isolation, right? But we have especially I think in healthcare systems like Sweden, where at least if the stories from the outside are to be believed, your healthcare system is much more organized than most places in the world. And so you do probably have the ability to use machine learning and AI models to look at the full picture in a way that other countries just don't necessarily have that luxury because they haven't set it up so effectively from the beginning. Is that accurate? Do you have a really strong national health data infrastructure, or is it just as messed up as everywhere else?
I mean, I'm not sure if it's as messed up as everywhere else. There's definitely improvements that can be made. We haven't really started using AI at scale yet, but hopefully that will become possible. And I think people are looking into it. And maybe, I mean, they kind of have been promising us in healthcare that with new electronic systems, our life will become easier for at least a decade, and it's only become more and more admin for different doctors and harder to make the correct interpretation calls. But I think that this will likely will become easier in the upcoming couple of years. But then we will have new difficult pieces that even AI can't solve. There will always be some sort of a line where we have to assess whether something is medically actionable or not actionable. But yes, we have a good healthcare system in Sweden. There are some issues with it being, for instance, regionalized, which makes it somewhat problematic to collaborate between regions, and you need all the legal requirements to be in place. But
makes sense. As we wrap up here, I want to just your research base is really broad. You're covering so many different therapy areas, lots of different technologies. I think the common thread is using advanced genome sequencing and genomic technologies more broadly to improve the way we do translation of genomic medicine. I want to understand what your big research priorities are for the next couple of years. What are you focused on right now? If people follow you, keep track of your work, what sorts of things are they going to expect to see?
So I'm fascinated with structural variants, and I've been sort of pushing into these different technologies on the research side because they really need better technology to fully understand the complexity of structural variants, to fully understand their biology and how they form and why they form and how they cause disease. I think with the new like reference genomes, the T2T, the Pangenome project, we really, to get coupled to like ultra long sequencing, we can really move into areas of the genome, such as centromeres, that we are realizing they are not just, I don't know, static, identical entities across people, but really dynamic and possibly could explain why some chromosome 21s can be stable in a trisomy state, or even in a marker chromosome or a cancer mediating rearrangement. So I think that this is really interesting, that we can kind of go back to the cytogenomic side. And I would love to see a true digital karyotyping where we make use of not only like the readopt but really looking to the split reads and try to resolve rearrangements in an individual genome and use de novo assembly to really have phase T2T genomes in our patients. I think that would be a step forward. But at the same time, then using this phase data to call variants and cis or trans and understand a little bit more about the total burden of the patient's genetic composition and how that links over to disease.
Yeah. Amazing. Well, that's very exciting. Anna, thank you for taking the time to join us and offer all the great perspectives and share your work.
Thank you for having me.
Great. And as always, thank you everybody for listening, and we'll see you next time. Thanks as always for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.